This is Goddess of Science Biology, and I'm Mrs. Wolf, and today we're going to talk about enzymes and the pH scale, which is the fourth part of my biochemistry series. So let's talk a little bit about enzymes. They have the ability to change their shape. So what happens is the reactants cause the enzyme to change their shape when they fit into a specific enzyme at an active site. The active site of an enzyme is very specific for the reactants that fit in there. It's kind of like your house key. Your house key fits into your house. My house key won't fit into your house lock, the lock on your house. So it's kind of like that with reactants and the enzyme that they're supposed to fit into. So here's a picture. We have this enzyme here, and it has a couple of active sites. Enzymes are very, very specific for their shape because these little puzzle pieces have to fit in here just right. Part of the job of the enzyme, as it lowers activation energy, is to bring the parts of the molecules together so that they can in fact react. So once they're fitted in there, they make something called an enzyme substrate complex. The reaction occurs, and then the products are kicked out, and this little knob that was on the green blob now is attached to the yellow blob. So those are the products, and notice that the enzyme is exactly the same as it was when it began. So once the substrates bind to the enzyme, it makes something called an enzyme substrate complex, which eventually falls apart into the products, giving you the enzyme back to be recycled and the products that you want out of the reaction. So if the active site on an enzyme changes shape radically, that would be like you go to school, you have your house key in your pocket, and you come home and your parents have changed the locks on the house, and so you can't get in. That would be what would happen if the active site, the shape of the active site, were changed radically. So the catalyst, the enzyme, is no longer going to work because the specific substrates are not going to be able to fit in there where they're supposed to. That when we talk about changing the shape of any protein at all, it's called denaturing the protein. It's inactivating the protein, or the enzyme in this case, by changing the active site shape. Some ways that you can denature a protein are pretty easy. You can have heat. You probably have fried an egg sometime in your life. Before you fry the egg, you crack the egg into a bowl, and the goo around the yolk in the middle is clear. As soon as you put that in a frying pan and heat it up, that goo is no longer clear. It turns into a white solid. That's an example of denaturing that protein. Strong acid or base can also do the same thing. When I was growing up, my grandmother used to make this really awesome chocolate cake, and you would have to put some curdled milk in there. Milk has a protein in it called casein, and we never had any milk around our house that would go bad because we drank so much of it. So she would to make this cake, she would take a cup of the milk, regular old milk that we had, and she'd put 2 or 3 tablespoons of vinegar in it, and then it would get that lumpy texture. Now you're like, oh my God, I can't believe she put spoiled milk into a cake, but it was delicious. So strong acid or base can also denature, and other substances can denature proteins as well. So moving on to our second topic today, pH. pH is a measure of the amount of hydrogen that's in a solution, not just in living things, but any solution at all. An acid, and these little bracket things are called they equal concentration. So rather than saying the concentration of hydrogen, you just write these little brackety things. It's very commonly used in chemistry when you're talking about concentrations, and it sure beats the heck out of writing out concentration every single time. So I'm going to use the brackets. You're certainly welcome to use the bracket as well. So if you have more hydrogen ion concentration than OH, which is another ion called hydroxide, if you have a greater hydrogen concentration than hydroxide concentration, it's going to be considered an acid. And putting that on the pH scale, normal human being would have done like 0 to 10. No, the pH people did it 1 to 14. Weird. So anything that has a pH of less than 7 would be considered an acid. Here's a nice little chart that shows you some things that are acidic. Battery acid is probably the best known. Stomach acid is very, very acidic. Lemon juice and vinegar is down here at a 2. Orange juice and cola is at a 3. Tomatoes, bananas, and normal rainwater is at about a 5. Healthy lake water, urine is a 6. So all of those things would be slightly acidic. One of the things about acids is they kind of are like sour tasting. But don't go around and like taste, battery acid, because you're just going to have to go along with me on that one, that it does taste sour if you could taste it. So some examples that you might come across is lemon juice, a tomato. When you're making spaghetti sauce from scratch and you're using fresh tomatoes, if you don't cook it for a long time to break down that acid, one of the tricks that you can do to kind of get rid of that harsh acidic taste of the tomatoes is you can put a little sugar in there, and that will kind of help it out a little bit. So if you're not acidic, you might be basic, and that is kind of just the reverse. It's when you have greater hydroxide concentration than hydrogen concentration. And that means that the pH is going to come in the register of greater than 7. So some things that are greater than 7 are seawater, baking soda, the Great Salt Lake if you happen to be in Utah, household ammonia, that's the stuff that is often in window cleaners and things like that. Soapy water is a base. We have oven cleaner and sodium hydroxide, which we talked about hydroxide concentration. Sodium hydroxide is very, very, very strong base. Some things that you might run across that are a little bit that are basic are human blood is slightly basic, is about 7.45, just slightly basic. And oven cleaner, back in the day before they had self-cleaning ovens, you would buy this stuff which in essence it was sodium hydroxide in spray form, and you put it inside your oven and you'd clean it out, and that was incredibly basic. So a neutral solution would have hydroxide and hydrogen concentrations absolutely equal. Pure water, and I'm not talking about water out of the faucet, even if you have a well, it's probably going to have some solids and other things dissolved in there. It's going to change the pH. If you take any water sample at all, boil it, collect the water vapor that comes off, and then condense that water vapor, that is distilled water, and that will have a neutral pH. The hydroxide and hydrogen ion concentrations will be exactly the same. And the neutral is pH of 7. So distilled water, I just described how that process is done, is considered neutral solution. Tears, kind of, it's a little bit more toward the basic side. Blood also, as I told you, is a little bit on the basic side, but pretty close to neutral. Reactions in a cell need to happen in a very specific pH range. It can't happen, like I mentioned, that blood is 7.45 to 7.55. If it's down closer to 7 or even in the 6 range, then bad things are going to happen in the cell. The reactions can't happen that need to happen. So they have a very small range. Most cells need to have reactions happen between 6.5 and 7.5. So not a big, huge window there. If the pH is out of the range, the enzymes won't work and we'll get into a denaturing situation with that. Body needs to keep the pH in range. Now you're constantly, eating acidic and basic foods, and all kinds of things are happening in your body that changes your blood pH. So your body needs a lot of mechanisms on how to keep that in that range. So as soon as you drink like a soda and it starts to be absorbed, the acid starts to be absorbed into your bloodstream, then your body reacts to that. It's one of those reacts to stimuli things, like a characteristic of life. So your body has chemicals that are called buffers in there. It's kind of a broad category. There are many different types of buffers. And what a buffer does is it resists changes in pH. If you drink a whole lot of soda, which contains phosphoric acid, once the acid gets into your system, the buffers are reactions that occur to change that chemical so that it doesn't cause big swings in your blood pH. So buffers are really, really your friend. They resist changes in pH, help to maintain homeostasis. Okay, so next time we're going to talk about macromolecules in part 5 of our biochemistry series. Thanks for joining me. Have a great day.